Hello, good morning, everybody. Welcome to the webinar. We're going to wait just a minute for everybody to join here. Oops. Good morning, everybody. We're going to go ahead and start because we have a few announcements as people trickle in. So the first slide is just an announcement of the speakers today. We have Scott Meschke and Edwin Oh speaking. And then we also have next month two great speakers. We have Rachel Porteski and Irene Zagararaki, and I'm probably pronouncing that wrong, and I will ask her how to pronounce it when I see her in June. Just a reminder of how the webinars are organized. The speakers will give 20-minute talks, and you should put your questions into the Q&A, and then we will have a moderator ask the questions, and we do have the ability to elevate a participant to a speaker if there's a question where there should be some more discussion. Before we start with Scott's talk, we have two other announcements, and I'll let Sarah give the two announcements.
Yeah, so the first one is we are starting a journal club. The first one is on May 25th at 10 a.m. Pacific time. Hoping that this is an opportunity for students and early career scientists to talk critically about some research that's out there, as well as network and meet other people who are doing the same work. The hope is that this will meet roughly once a month, but I'm hoping that it is a little bit led by the students who are participating so that we can help foster that. So just scan this QR code to register, and I'm looking forward to seeing you all there. And then the next one, the National Institute of Standards and Technology is working on a standards document for concentration of wastewater. And they have asked us to advertise this. If you scan this QR code, you can go and register to be on their email list, as well as access the Google Doc. They have a couple more meetings this month to really work on that. And they have invited everyone to participate because they want to make sure they have as many people with eyes on this document as they can.
Thanks. We'll also put that info in the chat. So remember to put your questions in the Q&A for the speaker. So Scott, you can go ahead and share your slides and begin.
Right. Is everybody seeing that okay?
No.
No?
Have you started sharing your screen?
I did. Let's try again. Failed to start Windows Connect. That.
There we go.
I get us there? All right. Hopefully that works for everybody. I'm going to talk relatively quick because I've got a fair amount to cover here. I built in a few extra slides for some unknown reason. Appreciate y'all for joining today. It's great to see so many people this early turning in for things on wastewater. I did take a quick look at who all was here, and I noticed a few CDC faces, but not a lot of folks from the polio group. So if there's anybody from the CDC polio group, please feel free to reach out and correct me as I'm going through here. So I'm going to talk a little bit about just some global perspective on surveillance for polio and wastewater. Surveillance for polio has been ongoing really since the early 2000s, and it's really a program that has pre-existed as part of the Global Polio Eradication Initiative, which we'll talk about in a moment. First, I want to introduce poliovirus because for a lot of folks, probably most on this call, they really never had any experience with polio. You know, back in the 50s and before, polio was a very serious threat, even in this country. And globally, it's been a threat up until just recently in many countries. We are fortunate that we are nearing eradication, and we're down to only 2 endemic countries and only 1 that's actually showing cases this year. But again, we'll revisit that in a moment. First, a little bit about poliovirus. For most of you, if you've heard of polio, it probably came from working with it during your doctoral or master's studies, because it's one of the more well-studied viruses that's out there. It's an icosahedral virus, about 27 to 30 nanometers in diameter. It has a single-stranded positive sense RNA genome that's about 7.5 kb. And its protein code's kind of interesting because it comes from a single large protein that gets cleaved into 4 capsid proteins that then fit together in a crystalline structure. This is distinct from some other viruses in that it is non-envelope and can be very stable in the environment. As I mentioned, polio is a member of the genus Enterovirus, and within that genus, there are 15 species currently identified. That includes enteroviruses A through enteroviruses L, and rhinoviruses A through C, and it's the enterovirus group C species that includes the poliovirus, all 3 serotypes. So polio is known for having 3 distinct serotypes, types 1, 2, and 3, and components of each of those are what we've included in the vaccines up until recently. Advancing. There we go. Polio, as far as the disease that it causes, can be quite disfiguring. Some of these pictures are fairly disturbing. I swiped these last night from the CDC website, just showing some of the deformity and limb deformity that's associated with poliomyelitis. But then also pulling from the AP a picture of a Boston polio ward showing that iron lungs, which really probably nobody on this call has ever really seen, just filling an entire ward with people actually inside. And these were essentially the only thing that was keeping them breathing. The interesting thing about polio that really facilitates environmental surveillance as a tool is that most people that get infected with poliovirus don't have any symptoms. Really, it's only about 1 in 4 that show up with some type of flu-like symptoms, and only a handful actually develop a more serious outcome like meningitis or paralysis. So meningitis, it's about 1 to 5%. Whereas with the paralysis, it's maybe 1 in 200 to maybe 1 in 2,000, depending on the serotype that you're infected with. What this means is that there are a lot of folks out there that are either asymptomatic or are showing up with very mild symptoms that are hard to distinguish from other viral pathologies. Again, because of the disfiguring nature and the fact that this was really actually killing a fair number of people, there was a big push to add polio towards the eradication list. And that stemmed this idea of the Global Polio Eradication Initiative, or the GPEI. And this is a public-private partnership that includes 6 core partners, including the WHO, Rotary International, CDC, UNICEF, and the Bill and Melinda Gates Fund, as well as Gavi, which is the Vaccine Alliance. And those 6 partners have really made it their ultimate goal to eradicate polio worldwide. So far, they've enlisted over 20 million volunteers, are active in 200 countries, and have managed to vaccinate over 2.5 billion children. I want to kind of distinguish between eradication and elimination. So eradication is a term that gets thrown around a lot with polio and actually a few other diseases now. But there's really only been 2 diseases that we've managed to eradicate. Eradication means complete elimination from the face of the earth, no more transmission at all. Whereas elimination is essentially ceasing transmission in specific geographic locales. And so polio is not eradicated, although it is on the edges. It has been the target of eradication since 1988, when the World Health Assembly passed a resolution, and that ultimately led to the formation of the GPEI. Okay, that step alone helped us get to a point where we've decreased poliovirus circulation and transmission by over 3 logs, 99.9%. Okay, it does not mean it's been eradicated. It has been eliminated in several continents, but it is still endemic in, like I say, 2 countries. The arm that actually does the surveillance for the Global Polio Network is called, or excuse me, for the GPEI is called the Global Polio Lab Network. This was established in 1990 by national governments and the WHO, and specifically was looking at AFP, okay, as the kind of whether polio was the cause of this acute class of paralysis and delineate it from other causes of paralysis. AFP is kind of the hallmark sample, but as you saw from the statistics, it's really only about 1 in 200 to 1 in 2,000 people. So again, that points towards a lot of people showing up infected with polio, but not showing the kind of hallmark symptom that a lot focus on. The lab network consists of 146 or so accredited polio laboratories in 92 countries in all 6 regions of the world. And then it processes, this is a staggering number of samples, 220,000 stool samples from AFP cases and more than 8,000 environmental samples or sewage samples every year. Now, that may not seem like a large number to some of you who've been cranking through large numbers of COVID samples over the last couple of years, but keep in mind, polio is different, and how we analyze polio is different. And so we'll go into a little bit of that in a moment. So let's talk about environmental surveillance for a moment. The key point I want to make about environmental surveillance, and that's what we call it. I mean, a lot of people talk about the term wastewater-based epidemiology, sewage surveillance, wastewater monitoring. All of these terms get kind of interchanged a bit, but really it's the surveillance piece is the ongoing and systematic sampling and analysis piece. And it is not a measure of exposure like many of the other types of monitoring that we do with wastewater. Rather, it is a supplement to clinical surveillance to understand the circulation of a disease within a population. And when you're conducting surveillance, it implies that there's some type of threshold response, right? Surveillance is done to further public health. It is a public health tool. And so that's one key point I want to make about environmental surveillance is that it needs to trigger an outcome if you get positives or if you increase past certain thresholds. Okay, so this isn't a new idea necessarily, but most of the efforts up until the polio program were largely based on research or local monitoring, outbreak investigation, but were not truly systematic environmental surveillance programs. The roots for environmental surveillance, particularly for poliovirus, but really overall, the only thing that was really prior to polio was some initial work on typhoid back in the 20s. But the roots for polio trace back to the 1930s with 2 specific papers published in the Journal of Experimental Medicine. The first actually worked on, I think, 1939, showed poliovirus in stool, and the second showed poliovirus being isolated from urban sewage. Now, in this picture, you'll notice a few folks flagged here, Albert Sabin and Jonas Salk. You might recognize their names because they are associated with both the live attenuated and the inactivated vaccines for poliovirus. But a couple other folks in this picture are just as important, particularly in regards to the environmental surveillance, and that's John Paul and Joseph Melnick. And they were the ones who were able to isolate polio from stool, demonstrate that it was shed, and then could be monitored in urban sewage. That led through a series of steps in development of methods and trying to understand what that meant in terms of outbreak investigation and others, to a true tool being developed in the form of environmental surveillance that gets used by the GPLN. GPLN started initiating environmental surveillance on a systematic basis back in the early 2000s. And by 2018, they had only 44 countries involved in this program. Okay, and so that was ongoing really at the beginning of the COVID pandemic. That was ongoing surveillance that already existed. They do both a systematic kind of regular surveillance at established sites, but they also do ad hoc sampling whenever there was any type of outbreak or clinical case identified. And again, the overall goal of this was to supplement clinical surveillance. So with this, I thought the next step that would be the easiest way to really kind of help distinguish polio from what the environmental surveillance that you're more familiar with, likely for SARS-CoV-2, was to look at distinct differences between environmental surveillance for polio and environmental surveillance for SARS-CoV. The first of these is the virus itself. And as I mentioned before, polio is a non-envelope virus. It's a pretty simple construction. You'll see up there, it has the 4 capsid proteins that fit together in this nice crystalline structure. It is considerably smaller. If you look at the SARS-CoV down below, it's about between 2 to 4 times the size. It's a little bit pleomorphic in terms of its overall shape. Still shows up mostly round, but has these distinct capsid or spike proteins on the surface. And it is an envelope virus in contrast to polio. Notice no surface projections of any significant size with polio, and no lipid component in its outer membrane. The second piece is also regarding the virus, is that poliovirus has different types that we have to consider, and we've defined these different types very specifically. The first being obviously the wild type virus. Okay, that's wild type poliovirus. That's the one we're really worried about when we're trying to eradicate it from the wild. But there's also something called a CVDPV. And the issue is that polio still uses an attenuated live vaccine that is made up of the different Savin strains. Okay, so these are vaccine attenuated viral strains. And at some regularity, these strains will actually revert and lose their attenuation and cause disease. And if they manage to circulate within a population for some time, they can actually cause disease and pop up, causing cases of poliomyelitis or AFP. And so we need to be able to capture each of these types of viruses to really understand the efforts to eradicate or eliminate polio from a particular region. I'll mention why that's important in a moment, but largely you have to keep in mind that when we're looking for polio, all these other types besides the wild type are going to be present. And so in some sense, we're always having to kind of tease out the needle in the haystack and characterize the different strains of polio. The next key piece that I mentioned is prevalence. And it's very clear, polio is on its last legs, whereas, you know, we've seen this immense drop in the number of cases from 1985-88 when we started the eradication initiative, and we saw really a logarithmic decrease, and now it's pretty well flat for the last 15 years or so. The issue is it's not as flat as it looks, and you actually do get numbers of cases, particularly in 3 key areas. This were the 3 countries that were endemic. Now it's just these 2 in yellow that are endemic. We do see occasional other importations of polio from these regions due to travel, but then we also see these CVDPV cases popping up. And if you contrast that with SARS-CoV-2, it's pretty much ubiquitous. It's hard to take a sample now and process it and not be able to detect SARS-CoV-2. But when you're looking for polio, you're looking for the rarefied fraction. You're looking for something that is not supposed to be there. So anything you detect is bad. You don't necessarily need a quantitative result with polio to have it be useful. Here is the most recent data on polio cases, and just shows you how close we are to actual eradication. As I mentioned before, there are 2 countries that are endemic, Afghanistan and Pakistan. And of those this year, as of this year, we've only had through May 1st, 1 case of AFP reported. That is extremely good news. You can see the number of cases have dropped significantly since 2019. And you see that we haven't seen cases in any of the other countries, no cases of importation of wild polio. What you can also see is that for these endemic countries where they are reporting their environmental results, you can actually see that they are still getting detections of wild poliovirus in the environment and from other sources, meaning the other sources could be healthy children that they sample as well, or other contacts that are not showing disease or AFP. But majority of these are really environmental cases. These are showing up in wastewater regularly. So we still know that the polio is circulating in those communities. That's just the wild poliovirus. If we start to look at the CVDPV, it's a much different story. And you can see 2023 is still better in terms of the AFP cases and also for the environmental isolations relative to the prior years, but still considerably more cases than what we're actually seeing with the wild type virus. Okay, and again, these are viruses that have reverted from their vaccine strain to cause disease again. Okay, you can see there are three types, the three serotypes: polio 1, polio 2, and polio 3. Polio 2 seems to be the most susceptible, and in fact, as of April 16th or April 2016, they actually pulled type 2 from the vaccine, the trivalent vaccine, and now only use a bivalent, and will soon be moving towards a monovalent PV1 because PV3 has been absent from the wild type for quite some time as well. PV2 has been absent from wildlife for a couple decades now, or not from wildlife, from the wild type has been absent for a couple decades. The third key difference here is the use case. And polio is really used to monitor this silent circulation, monitor progress towards eradication, so reducing caseloads, but it's also used to monitor the use of the vaccine, and it's used to look at the disappearance of the vaccine strains following cessation of the vaccine. And I'll show you a couple of examples of that in just a moment. It's also used to better assess elimination in cases of outbreaks. So occasionally we get these importation outbreaks or a VDPV outbreak, and they will do ad hoc sampling to kind of understand the containment around that outbreak. And then, of course, we want to document transmission importation. There are some cases of countries that are not in a at risk from having the CVV typically because they're no longer using the oral vaccine, but they do risk the potential importation from countries due to travel of people from regions still using the oral vaccine. And that was pretty clear when we got our first AFP case from polio in the US this last summer or so up in New York. And that led to the detections in the wastewater there as well. Of course, that was linked to ongoing outbreaks in Israel and in London as well, just due to a particular community that had spread due to travel. So here's some data back 2019, 2020, where we were looking using a couple of different methods for wild poliovirus. And I just want to show you that we are still able to detect this in the environmental samples. Each of these are established sites. There are now 76 sites, I believe, established in Pakistan. But these were just the subset. We were doing a method comparison between a method we developed, the BMFS, and a two-phase method. And what we were able to see here is that both methods are capable of pulling out the virus with near equal efficacy. There might be a slight preference for one method over another, depending on the type of site it is. For instance, here in Multan, the BMFS was performing much better than the two-phase method, but in other places, the two-phase method was detecting. And so this is the wild virus that we're able to pull out of the environment. The other pieces that are interesting, I'm not sure if that's not going and not advancing.
Scott, sorry to interrupt you, but we're at time now. So
Oh, shoot. Okay. Happy to stop. You want me to unshare and ask any questions or?
yeah, we can do that. We can do questions. Or if you have one more slide.
Let me give one more if I can actually get it to forward, but it's not. There we go. All right. This is the key. One of the key ones I want to show you is that we can actually track individual strains as they start to disappear. And you'll notice these ones in red are the ones where the Sabin type 2 were used. And this long red line here was where the actual type 2 virus was removed from the vaccine. And so we can actually see the numbers of viruses start to stop their circulation or cessation associated with the vaccine. And again, both methods worked quite well for identifying that. Okay. And we were still able to pick up the CVDPVs later of the type 2 once they came back. So I'll go ahead and stop share and address any of the questions. There were a couple other points I think are important to look at, but we can chat about that.
So I guess the first question, there was a request to talk about the difference of the biocontainment between polio and other things that people might be working with. I think you probably had slides on that.
That was my last slides. Yeah, so the last big difference between polio and SARS-CoV or other targets is that because it has been part of this very established, very well managed environmental surveillance program, there are differences for containment. As I mentioned before, environmental surveillance should have a threshold trigger. And for polio, that's really going in, doing additional sampling, and then initiating an SIA or supplemental immunization activity. And so the other big difference is that we tend to do live virus monitoring, or actually put it into tissue culture, rather than do PCR direct detection with polio. And part of that is because, one, it does increase your sensitivity in terms of the amount of volume you're able to assay, and two, it allows better characterization. And there's something called GAP3 going to GAP4 now, which is the Global Containment Program for polio. And that means basically you have to register as a polio active lab. You have to get audited and make sure that your space has adequate control and containment, both for biosecurity as well as the biosafety levels, which have been ramped up. That really complicates you processing other samples.
Yeah. So there's a question in the Q&A as well about if you could talk about the recent AFP cases that are derived from the NOPV.
Yeah, so I can't say a lot about that one because I haven't really read up on it. But the NOVP or NOPV is a new novel set of strains for the vaccine, and it was intended to really minimize this reversion back to a pathogenic state. And in fact, I think the evidence has shown that it still is, but they did still get limited reversion, and it resulted in a couple of cases of AFP. But I believe, compared to Saban strains, that it still shows like it's got less reversion.
And then there's another question about if the data presented are from the full culture-based method, and I wonder if you could talk a little bit about what that process is with the whole culturing.
Yeah, so that was one of the other slides, was regarding the methods. The methods here are very different. Whereas there are multiple concentration methods, there is a recommended method that is a two-phase PEG dextran method that WHO uses. But there are other methods like the negative filter method or a positive filter method, BMFS, that are acceptable methods for concentration. So similar to SARS-CoV in that there are multiple concentration methods, but then it goes on to tissues instead. And there's an algorithm they run of 5 L20B cells, which is a mouse cell line that's been modified with a polio receptor, and 1 RD cell line flask. And they look for monitoring growth in either of those flasks and then cross plate them to see whether or not they get growth. And then they go in to do PCR for intertypic differentiation.
And then there's just a very basic question as well about the level of biosafety that is necessary for wastewater work with polio.
All right, loaded question. Technically, if you are working with wild type or you were working with any type of type 2, I believe you're supposed to be at BSL-3 now, or 2 with 3 practices minimally. Realistically, most people have been working with a type 1 LSC and have been used to working with it at level 2. That was adequate biosafety just a few years ago. And really the ramp up is considered, I think, largely just to further the containment piece. Okay, it's much like smallpox. Smallpox used to be a BSL-3, now it got bumped to BSL-4 when they eradicated it.
Great, thank you. That's all the time we have for questions. Thank you so much. So now we can hand it off to Edwin.
Hello. All right, let me try and share my screen.
Perfect.
Let's see. How does that look? Do you see the right screen?
We see the speaker view.
Got it. How's that?
It's a little small, but we can see it. I don't
Okay.
know if you have any like font that's small, but it should be.
Okay. All right. I think. Okay. All right. So thank you so much for the invitation to speak today. It's a real pleasure. As a human geneticist, I often tell all my colleagues that I've been so fortunate to work with a new group of individuals who've educated me so much on the beauty of wastewater biology. Um, a lot of our work is supported by a host of undergraduate students, like many folks on this call. I think a lot of us have developed our programs knowing that when we go out to various locations for collecting samples, none of this would be possible if it weren't for the fantastic group of students that a lot of us have at academic institutions. And so I always like to start my talks with acknowledging the folks who actually do the work, because I go on and on and on. And the reality is sometimes I'm not as fortunate to be able to collect samples, and I end up writing the grants and writing the papers. But here are really the individuals who've done most of the important work. And a lot of what I'm going to share with you today is driven by a talented junior faculty member in the lab, Dr. Van Vo, and our undergraduates. As I mentioned at the beginning of the talk, and again, thank you so much for the invitation to speak. We ended up working with environmental engineers, computer scientists, and a number of epi folks in being able to understand this beast of SARS-CoV-2 that causes COVID-19. And today I'm really going to talk about how we've used our sequencing approach to be able to understand the evolution of SARS-CoV-2 and how we use this to pivot to antimicrobial resistance. And I'm going to actually talk a little bit about Mpox, because some of our panels for antimicrobial resistance weren't at a place where I felt that we were ready to share at this point. But I'm going to discuss a little bit more about why and how I think some of this could be done better using these various pathogens as examples. None of what I'm going to share with you today, again, would have been possible if it weren't for our collaborators and our students. Our main collaborator is Dr. Dan Garrity. He's taught us everything about environmental engineering, from what a manhole cover looks like to, unfortunately, sometimes I have to admit what even a crowbar looks like. And his postdoc, Katarina Papp, and our epi folks up here, Dr. Lockett, Dr. Khan, and Ms. Franek and Mr. Zhang. A lot of what we do is a public-private partnership between various state agencies, universities, and commercial companies. It's been a fantastic collaboration to work with a number of these different private companies in developing better panels to deal with this complex matrix such as sewage. We're also funded by a number of different organizations, and they're listed here, including NIH and CDC. So we set up a dashboard for the state of Nevada, and we ended up collaborating with a number of different universities and organizations in setting this up. It's hosted on this website right here. And in this website, we have data for the state of Nevada. The state of Nevada is made up of about 3 million people. 600,000 or so live up here in Reno and Carson City, and 2.4 million people live down here in Southern Nevada. And so if you hover over these icons and you click on them, you'll get a sense of how viral counts have evolved over time. We focus a lot more on Southern Nevada with our surveillance, mainly because of geographic restrictions. We work with Dan in looking at the sites that service the urban areas in Southern Nevada, including Las Vegas and Henderson. And we also look at rural sites out here in Indian Springs, in Moapa Valley, in Blue Diamond, Searchlight, and Laughlin. And again, we can see from these graphs down here how viral counts listed here in green have changed over time relative to case counts shown here in gray. And these numbers are generated from dates starting back in 2020 till today. Now, when we look at Southern Nevada, some of you may recognize this image. Southern Nevada, the urban sites are serviced by a number of different sewer sheds here, numbered from 1 to 6. And many people, when they come to Las Vegas, show up right here where the star is, and that is the Las Vegas Strip. That area, the Las Vegas Strip, sees about anywhere between 800,000 people to 1 million people every week. And all of that waste goes towards this sewer shed here that services the Strip and also the local residents in the area. So as I mentioned in the beginning, I'm really going to focus more on how we've approached our wastewater monitoring programs using NGS, or next generation sequencing. For the most part, we began our sequencing protocols using something like 10 nanograms of cDNA or DNA, but we've come to realize that this is a very loose standard of a starting material, because when we talk about 10 nanograms of cDNA or DNA, this is from total wastewater samples that haven't necessarily been depleted of bacteria or fungi or viruses. So this has, again, been rather loose in our interpretation as we move forward. I think the biggest difference for us hasn't really been the amount, but rather the quality of the nucleic acids that we're able to obtain from the get-go. So we've worked with a number of different companies and looking at hybridization-based and amplicon-based kits. And what we ended up doing was working together with Paragon Genomics in looking at sequencing of SARS-CoV-2 in mpox. And more recently, I think what we've ended up doing is we've switched completely to more of a custom approach, mainly because of the uniqueness of wastewater samples and the nucleic acids that we're able to extract. Many genomes that we've been looking at are relatively small. And when I say relatively small, I'm comparing this to a human exome or human genome that we've been sequencing previously. And so for us, a NextSeq 500 or 1,000 has been more than enough real estate space on a flow cell to be able to look at anywhere between 100 samples to 1,000 samples at any one time. And what we've come to realize is that we need to achieve about a 50 to 100x depth over enough of the genome to be able to make calls about variance and variation as we look at the different pathogens. And I got to tell you, just reading a lot of the literature that many folks on this call and in the community have published on sequencing has really helped us as we've developed our approach in looking at sequencing these different pathogens in a high throughput manner. So I'm going to start with a very, 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 very old story. We looked at sequencing of alpha B117 or the UK variant. And a lot of times what we end up doing is look for mutations that identify the unique variant that we're looking for. And so we'll have a patient sample, or we'll have a synthetic RNA that we'll use as a positive control, just to be sure that we can, that our workflow is able to give us the information on variation that we're looking for. Well, back in the day, almost feels like a very, very long time ago, we worked with Dan and we were isolating nucleic acids to be able to get to this point in December 2020, when we could start looking at genomes. It took us a very long time to get to this point where we could cover anywhere between 70 to 80% of the genome at this 50x to 100x depth. And I think two of the reasons that we took this long was because we weren't getting the quality of nucleic acids that we really needed for this. And many library prep kits were going on assumptions that made a lot more sense for clinical samples. So what was the interesting finding for us in this story was that we were able to identify this variant as early as December 2020. And when we compared these results to sequencing of clinical cases infected by the different variants during this time, we worked together with the health district to run a lot of these different sequencing experiments. And so in December 2020, you can see these three different variants or clades, lineages that were described back then as 20A, 20B, 20C. And we didn't see B117 show up until January 2021. And we started to see alpha increase over time slowly in our clinical sequencing. Now, I think as we've thought about this more and more, and frankly, when we were sequencing a lot of these samples, we soon realized that the number of samples that we were sequencing really wasn't at a point that it needed to be. And many of these numbers now have a zero added to it as we, or two zeros added to it as we've looked more closely at samples that represent each month. But the main point here was that we didn't identify alpha until January of 2021, but we were able to identify alpha almost a month ahead. So this is fairly consistent with, I think, a number of different folks who've shown something similar to this. And we've moved forward and we've asked ourselves, did we get lucky, or is there really some precision in wastewater sequencing that allows us to predict variants in a community? And I've been really pleasantly surprised that whole genome sequencing of wastewater samples have really helped us predict the emergence of a number of different variants, sometimes pretty much on par when they show up in the population, and more recently, sometimes as early as 1 or 2 months before we have the first recording of a human infection. So this was back in 2020, 2021. We wanted to look a little closer at the evolution of Omicron with our tools, and we were able to then compare Omicron as it was developed as infection numbers were increasing over time in 2022. This is an image of sequencing of clinical cases where in January, we're showing data from January to March to April, all the way to June. And like, again, in many of these figures that I think a lot of you have seen, in clinical cases, we've seen BA1 be displaced by BA2, then 212.1, and the other variants in early 2022. We wanted to compare these results when we were sequencing at scale for clinical samples, 1,000, 10,000 samples versus wastewater samples, where this bar is representative of one single sample isolated in March from a single wastewater treatment plant versus 10,000 different samples that was representative of a single week. And again, this is data from a treatment plant from January of 22 to June of 2022. And if you juxtapose the two different data sets together, this is what you can see. And the bottom line is that with wastewater sequencing, we can see that same pattern of displacement of BA1, 2, 212.1, and BA5. And this pattern of displacement is happening at a more rapid pace than what we see with the clinical sequencing. I think a lot of this has to do with our ability to get samples real time from these different wastewater treatment plants. I've been, to this day, still very, very surprised at how we can identify these variants much sooner in the wastewater samples. And so because we did this from January to June of 2022. We then obviously did this for the last 15 months. And so from January of 2022 all the way to pretty much the end of April of last month of 23, this is the type of pattern of displacement that we can see with BA1 to BA2 to 12.1 to BA5, BF7, BQ1, and XBB.1.5, and .16 and .9 for XBB have already shown up very much in March from our wastewater treatment plants, and a little sooner in some of our manhole samples. So we can also do this, since we can do this for one wastewater treatment plant, we can also do this for all of the different wastewater treatment plants. And the bottom line of this image really is that this follows the same temporal timeline that I was showing in the slide before. And you can see all of these different patterns show up at both rural and urban locations. And we can quickly track these variants a lot sooner in the wastewater than when we actually record them in humans in those corresponding zip codes. And so we've worked together with the public health folks to be able to show and share this information in anticipation of some of these variants as they show up over time. So having this experience working with the wastewater samples and with SARS-CoV-2, we felt that we could now look at other targets, other pathogens. And so we worked on this study back again in 22 to look at some samples that we had collected in 2020 and 21. And the question here that we wanted to ask was, could we now look at those samples and the extracted nucleic acids and look for more than 2,000 different antimicrobial resistant alleles that would match up to potentially resistance to antibiotics or classes of antibiotics during that time. And so the hypothesis going into this study was, well, if there was a surge during a certain time of the year, which corresponded to alpha, would we be able to see a corresponding surge of antimicrobial resistant alleles? And honestly speaking, the surge or an increase in the amount of antibiotics that we could detect in our wastewater, and at least at the genomics level, we're able to show that this was true for some antimicrobial resistant alleles that matched up to beta-lactams and fluoroquinolones. Now, I must confess that this was our first stab at being able to do this, and our process has advanced a lot more since this time to look more closely at the alleles and to get so much more depth for sequencing depth for these different markers. So with this experience again of being able to sequence SARS-CoV-2 antimicrobial resistance, as we were developing our tools for AMRs, Mpox hit, and we had to pivot to use our tools and work with our public health folks to be able to track, detect Mpox, and also to be able to sequence Mpox in clinical cases and in wastewater samples. And we wanted to do this at a facility level and at a community-based level, where we could look at shelters, hotels, conference areas, and universities. So like many other places in the country, Nevada was hit with a large number of cases of Mpox, and most of these cases were happening between June, July to October of 2022. Here is an example of the case counts that we were seeing in Southern Nevada during that time. In the x-axis, we have time, and in the y-axis, we have the number of cases. So every box here is an individual or 5 or 6. And you'll see that most of our cases were registered again during this time of July and August. And so we went back to the samples that Dan had collected, and we felt confident that given our experience, we were going to be able to detect Mpox, especially as it was emerging in July and August. And unfortunately, to our dismay, well, in some ways, because we felt that we weren't having the sensitivity, we weren't able to detect Mpox in the various sewer sheds until sometime in September. So in August, we were scratching our heads. Why were we getting 1 out of 3 hits or 2 out of 3 hits? Why weren't we getting the triplicates that we were typically getting for a pathogen that we were confident was present in the wastewater? So during this time when we were getting 1 out of 3 hits or no hits at all from the quantitative PCR detection of Mpox, we decided to go back to this strategy that we used for looking for Omicron as it entered into Las Vegas. Many of you are familiar with this site of the Las Vegas Strip, where we have many, many different hotels along this long corridor of tourism. And we've worked with a number of different folks, and we've always thought about the option, and we're not there yet, but of the option of being able to sample from each of these different hotels to get a better sense of pathogens in one specific location. So that was one approach, to be able to push that forward. The other approach was to break up the strip into various different zones. There are multiple sewer pipes that go through the strip, and then they swing east, such that we can collect information from zone 1, zone 2, zone 3, just by sampling from a manhole that's east of the strip. And so using this approach, we then went back to look at our case counts. Here is what we were seeing for our case counts. And finally, we found an area that had viral counts that was more similar to what we were seeing for case counts. And so we were then able to be in a position where we could take these samples and sequence these samples and track the evolution of these different clades that were emerging. And we obviously compared what we were seeing in the wastewater samples to what we were seeing in clinical samples. And here's a depiction of what we were seeing in clinical samples from June to October. And we thought, aha, we've got it this time. And to our dismay, as we worked with a number of different commercial companies, we were getting great depth of certain parts of the genome, but terrible depth in a lot more areas across the rest of the genome. So we felt like, oh my goodness, this was deja vu again 2 years later, and we weren't able to get the resolution that we needed. And this is when we started pivoting to more custom approaches. And then we finally came to a more custom approach where we can cover all 200,000 nucleotides, at least at 50 to 100x. At the 70 to 80% genome coverage. And now we can really track, we could really go back and track all of our different variants and show that there were a lot more of these various clades early on in time.
Edwin, sorry to interrupt you, but I wanted to ask you to wrap up so that we have questions before.
Sure thing. So ultimately, a lot of folks have asked us, why are we doing this? Why are we drawing light on these types of infections? And the reality is, I think by this approach, we were able to then work together with the public health folks and be able to direct the mobile vaccination teams to areas that could benefit from vaccinations. And we were really shocked here. We started setting up these clinics at bars and pubs with the highest levels of Mpox levels from 10 p.m. to 1 a.m. And, you know, I don't know about it, folks here, but when I was 20, 21, as I was going out to areas like the Strip, I wasn't thinking about getting vaccinated. But surprisingly enough, we got 30 to 50 first shots into arms and something like up to 50 shots or second doses from this approach that may not have happened. So finally, our new vision for NGS and AMR is really to be able to go in and develop our own custom approaches where we can look at these viruses, these bacteria and fungi, by taking into consideration that these aren't clinical samples. We've worked with a number of different folks with the panels, and they haven't really worked for us. I'm curious how the community has, the experience of the community working with the various different panels, but for us, we've had to go in and develop our own panel to be able to get the confidence. And how do I judge?
Sorry to interrupt you, but there's a lot of questions.
Oh,
and I
okay.
think that
Sure
those
thing.
are really fun to answer. So maybe we can just start questions now because we're really only have 4
Sure
minutes
thing.
left.
All right. So, and our confidence is really judged by SARS-CoV-2. Thank you. Happy to take questions.
Thank you. So I'm gonna just rapid fire these as much as possible. So one question that I had also was how you attributed differences in wastewater and clinical with respect to variant compositions. Could you attribute some of that to the tourists who may not be subject to clinical sampling, and how it affects how you interpret the variant composition for permanent residents in Southern Nevada?
Yeah, 100%, right? That's such a key point. And I think we've used this approach here to really help us better understand what is the contribution of tourism to the entire area here in facility 1. So we know that we can sample from the strip, from a manhole, and then we can compare those, the viral counts there and the variant composition there to the rest of the entire facility to be able to get a grasp of how variants are traveling into our community from tourism versus the local community. That's been one major approach for us to track this pretty difficult question to track.
Thanks. And someone was wondering if you've tried sequencing influenza.
Yeah, we ended up publishing a paper on a vaccine-resistant version of H3N2. And I've got to tell you, this was our first stab at looking at influenza. And we got decent depth around some parts of the genome, but we were so far away from where we need to be. And so I think today we're at a place where we can go in and get those metrics that I shared with you of about 50 to 100x depth across the different chromosomes for influenza, and not necessarily just some parts of the genome, which was really what we were restricted to for some commercial kits. And this is by no, I want to be sure that I acknowledge that the commercial companies here do an amazing job. And the goal here is for many of those kits is potentially to genotype these strains and not necessarily to cover the entire genome. So we were going in with perhaps the false or the wrong impression. And as we've developed our impression of what we needed, our custom kits have evolved as a result.
Okay. On that end, I guess, do you have any details or tricks that improved the performance of your custom library prep? And then someone asked if you're using the Swift IDT kit.
Yeah, we started with a lot of those kits. I think a lot of those kits have gotten better, but in the beginning, the best record of, again, depth and coverage was through Paragon Genomics. And I think it was because of their design of their amplicons. And we've used their creativity and their imagination as we've developed more of these custom kits for other pathogens such as gonorrhea, chlamydia, Candida auris, for some of these larger genomes that we're now working with. And
thank you.
And to answer the first question, it's really been the quality of the nucleic acids that have made a big difference to us. Not all kits are equal in our experience for sequencing.
Yeah. I guess we're out of time for now, but thank you so much for your presentation. It was wonderful to see all the sequencing, how you're leveraging sequencing for the monitoring process.
Thank you.
Thank you, everyone. Thank you, Edwin and Scott. See you all in a month. Bye.